early in the evolutionary timeline. So phylogenies, you can kind of think of them as going backwards in time, where, at the ends where there are the most branches, we can think of that as the present situation. And as you go back, you go further and further in the past. And so a species that's incredibly different from everything else is going to have branched away from them the longest time ago. And so this kind of denotes that period in time where species G separated from everything else, A through F. So the further back you go to find, that breaking point tells us that this species is more distantly related than everything else. So we've got A and B and G. Now we just need C, D, E, and F, which maybe isn't as obvious. But what species might we want to put here? C, right? Because now we're looking at kind of, on average, groups A and B, what is the closest related to that? Not necessarily just A or just B, but since they're about the same, it's pretty easy. But you would look at this group of A, B, what's the next closest related thing? And C only differs from them by one. How about these next three then? D? You got D, E, and F. Yeah, so we can look at it in terms, not necessarily of which one is kind of the closest related to G, because it's not very clear, right? So G, there are fewer differences between E and G than there are between D and G. But there's also fewer differences between E and G than there are between F and G. But, this doesn't really tell us, who, we can't necessarily tell which one of these is most closely related to G. The reason for that is because G branched away from everything else. So what we could say is if we had maybe a branch right here, that G, say this is a hypothetical H species, that G is more closely related to H than to everything else because it branched away from H at less time ago than it did from everything else. So we can't really say anything with this phylogeny about whether G is more closely related to C or D or E. That we can't tell. But we can relate, we can, for instance, say that D is more closely related to species A, B, and C than E is because we have these nodes to kind of give us that point. So basically what happened here was A and B divergently evolved, they speciated, so there was a common ancestor that broke into these two populations or species A and B. You go back a little further, C broke away from the group that would eventually become A and B, right? So this is, we can call it, species AB. And this is species C becoming species C. So again, we can't say when did C separate from A or when did it separate from B because there was no A or B. It was just the ancestor that would later become A and B. So we can say C broke away from AB a little bit longer. And then a little bit longer, what would become species D broke away from some population that would later become populations A, B, C. So it's not breaking away from population C, it's not breaking away from A or B, it's breaking away from the ancestor that would eventually become populations A, B, and C. Go back further, you just repeat the process, right? What would become E separated from a population that would become populations A, B, C, D, and then lastly, F broke away from a population that would give rise to populations A, B, C, D, and E. And then all the way back, and you're separating from G at this point. So Does that make sense about how we can interpret phylogenies? It doesn't necessarily tell us, you know, if G is more closely related to D or E, but rather how G relates to a group that would give rise to other species. So phylogenies there. What you want to be familiar with as it relates to phylogenies is interpreting those relative relatedness between species. So, you know, that comparison between species A and B, those would be kind of closer to the present when you're reading a phylogeny. So along, you know, closer to the end of the phylogeny, and then as you go to the root of the phylogeny, closer to the beginning of the phylogeny, this represents more ancient, longer ago breaks between populations. So these are all sort of hypothetical populations at this point, but what's interesting about this is that it actually represents real data, and specifically it refers to a gene in primates. That's sort of a nice little tidbit of information here, which is that this is all actually measuring relatedness between different primate species. According to one gene in particular called the GULO gene, it's just one of many enzymes, many proteins involved in synthesizing vitamin C. So what happened essentially is that many animals, including lemurs and non-primate species, synthesize their own vitamin C naturally. You might know because it's often kind of added to our diet, supplemented in our diets, that we don't synthesize our own vitamin C. We have to get it from milk and other nutrients. So we can't naturally synthesize vitamin C because of a mutation in this GULO gene. And it's not just characteristic of humans, but most primates in general. And what this phylogeny is showing is actually, you have the key here, but the main thing it's showing is that lemurs are very different than every other primate, every other monkey that we know about. So upon the evolution of Old and New World monkeys, when they branched away from lemurs, what happened is those ancestors that would later give rise to humans, chimps, gorillas, etc., developed a mutation in this gene, and they had to acquire vitamin C through their diet. Lemurs, however, they broke away before this mutation arose. They don't have a non-functional GULO gene. They still synthesize their own vitamin C. And so there's this clear difference between most monkeys and lemurs, not just in the way they look. You can kind of see that they look pretty different, but in their ability to synthesize their own vitamin C. So this is just kind of one of many genetic evidences that kind of falls in line with how we understand how we are related to other primates. And the GULO is just kind of one hallmark example where, lemurs are probably the furthest primate that we're related to, and we see it in a trait like vitamin C synthesis. They can synthesize vitamin C, and all the monkeys that kind of broke away from those lemurs developed that mutation, and we just inherited that since throughout time. We haven't gotten, another monkey evolve vitamin C synthesis since then. So just putting some actual context to this hypothetical activity here, and showing how we use this information, primarily genetic data, to make these phylogenies. So yeah, that essentially wraps up already the last real activity of Module 2. What I want to do now is give y'all the option if y'all want to essentially watch a little bit of a video right now, or if y'all want to get right into what I try to leave time for, which is just reviewing some concepts that maybe we didn't, that I think from making the exam are particularly important to know, or stuff that maybe we haven't had time to go over in general. So I'll leave it up to y'all. I do have the video up on Blackboard. This is really one way we can kind of, the video is meant to drive home this point of how we use genetics to make phylogenies. So if you're having trouble grasping this concept of phylogenies, which is a big part of Module 2, then this video is really helpful. But I'll throw it out to an iClicker poll. What do you want to do? I should just have this slide saved from now on. Do y'all want to watch a 15-minute video on phylogenies? Do y'all want to go over Module 1 and 2 concepts? Or don't care? Give y'all the option on iClicker to choose. Give y'all about a minute, and we'll go from there.
Are they both going to help us with the exam?
Yeah, for different reasons. This one, because phylogenies are the newest concept y'all have seen, this will elaborate on that. It is up on Blackboard, so if we don't get to it, we might get to both of these, right? Over the material I'm going to cover. It's just which do you want to prioritize or go through first. About 15 more seconds. Okay, most support therefore going over Module 1 and 2 concepts. So yeah, let's go ahead and do that then. The video is on Blackboard, and I've made a point of, it's an hour-long video, but I've noted kind of the two sections that are the most relevant for material that we go over, but it gives us a sense of how molecular data allows us to not only relate how we kind of first looked as humans, but also how related individuals are now using this approach of molecular phylogeny. So not just to compare distant species, but within a species, seeing how related we are, where geographically we might have come from. A lot of that is based off of genetic data. So what we'll do though now is kind of cover, I've gone over the exam, and there were just some points that kind of came to light that I want to kind of drive these points home, because maybe they're a little bit more represented on the exam, so good to keep these in your mind. The exam in general, most of the unit exams have a pretty similar structure. They're anywhere from 30 to 35 multiple choice questions. So that'll be on Scantron. Basically, you'll get a hard copy of the exam, and then just like an IRAT, you'll fill in all those multiple choice questions on the Scantron. And then there will be an 8-question short answer section. So everything can usually be answered in anywhere from 1 to 3 sentences. There are 8 short answers, but the way we kind of always do it, oh, it doesn't work. is the first four, so short answer questions 1 through 4 are mandatory. So you have to answer the short answers 1 through 4. And then 5 through 8, which is the next 4 short answers, you choose 3 to answer. So if there's one concept that you just know you're going to bomb that, you can do the other 3.
What do you mean choose 3 to answer?
What's that?
What do you mean choose 3 to answer?
Yeah, so for questions 5 through 8, that's a total of 4 short answer questions. You choose the 3 that you want to answer.
Okay.
Yeah.
And if you're ambitious and you want to go all 4, you get
extra credit?
for that 4.
No extra credit there, but I always have an extra credit question at the end. So
Can we
there
swap
is-
the extra credit one?
What? Yeah,
Can we
yeah.
swap the extra credit one?
Maybe. Yeah, you could, but yeah, I'll allow that. If you answer all 4, you can kind of do that instead of doing the extra credit. But you can't get credit for both, though. You can't do the 4 and the extra credit.
What about going above and beyond?
The extra credit is usually, a dumb question, a more creative question. So usually easier to answer, but maybe you've been studying the material so well that you just want to do, answer the 8 non-extra credit. You can get the extra credit that way. So that's the general structure of these exams. What I want to do is, as I was going through making the exam, there were a few points that I just want to drive these points home. The first is this difference between bottleneck and founder effects. So both of them describe instances of how you go from essentially what was originally a large population to this very small population. And remember the different mechanisms of evolution. Which do you think is going to have the largest effect in a small population? Which mechanism of evolution?
Bottleneck?
So remember what the mechanisms of evolution are. One of them is natural selection. What are other ways evolution might occur?
Natural
Adaptation.
So adaptations are what natural selection selects for.
Yeah.
So that kind of is lumped in natural selection. But there are other terms I'm thinking of here. One of them, genetic drift, gene flow.
Large population.
So large population kind of addresses genetic drift. What was the other one? So there's natural selection.
Random.
Mutation, right? Yeah, so mutation is that other one. So in a small population, this is really where genetic drift has the biggest effect. So remember the example of, I think I used the example of, a population of just 5 insects, right? How, genetic drift is essentially, a foot happening to squash some of those bugs, right? So the effect is going to be much more drastic if you're stepping on a population of only 5 bugs, versus if it were an entire ant pile where there's thousands upon thousands of individuals, and one stump of your foot and knocking out 3 individuals is not going to have much of an effect, right, on those allele frequencies. The bottleneck and the founder effect are essentially ways that that genetic drift can occur. So the way that I like to differentiate between these two is a bottleneck effect is usually going to occur through some form of disaster, right? So a bottleneck effect is something we saw in that Darwin's finches video. We actually saw both bottleneck and founder effects. So the first thing was a founder effect, actually. So what was this the timeline of the Darwin's finch story? What was kind of the first thing that happened before these birds could evolve into all these different species? So there was a drought, but long before this, what happened?
They founded the
So a bird or a small group of birds made it to the Galapagos Islands, right? So this is a founder effect. They founded a location. So whenever we talk about founder effect, it's usually an example like that, where a small subset of individuals make it to a place, and now you have a really small population in a brand new location. And the ones who make it there might be randomly assigned. So, we can imagine a scenario where maybe every year these birds are trying to just leave South America, where they originated, where the large mainland population was. So this is kind of South America. Galapagos Islands are these small islands here. And you have a big population of hundreds of thousands of finch birds. Just gonna make that the bird. And so every year perhaps there's migration going on, right? Maybe the majority of them just travel south to escape the heat near the equator, and the majority go there. But you can imagine some individuals are going to try and just go somewhere else. Maybe a small subset kind of go further inland into South America. Not as many here, but some here. Maybe some will even kind of go north, an even smaller fraction. Maybe some will even just try to do like humans in Europe did, right? Try to see what's beyond the sea. Can I make it there? So a really small fraction might try to do these journeys, and they kind of don't make it, they die. And the vast majority of them die. So we start off with this mainland population of maybe 1 million birds, 1% of which, so 10,000, try to make this voyage. 1% of that, 100, maybe make it all the way to an island, right? So now from 1 million, this population of 1 million, a very small subset, small group, make it to a new location. So we call this the founder effect. It's not describing a lot of death, right? Some maybe just don't make the journey and some die, but there's still a huge population here in South America. But a very small fraction, 0.01%, for example, just an example here, make it to this new location. This is kind of the epitome of a founder effect. Doesn't involve much catastrophe, right? Those instances in the examples that you read that describe catastrophes would be bottleneck effects. So this is like what species remained after the mass extinction of the dinosaurs. When the meteorite hit southern Mexico, it wiped out all the dinosaurs. And in a way, this was, on a global scale, a huge bottleneck effect. Because did every single dinosaur get wiped out? Yes. Not necessarily. It depends on your definition of a dinosaur, right?
Well, some evolved.
So one group made it, right? They became the birds that we see today. They evolved from raptors, from one group of dinosaurs, and they just happened to be the ones to survive. So what was once this large population of many species, a large community of dinosaurs, groupings of multiple species are referred to as a community, 99% of those species were wiped out, but a small group did remain. They became the birds that we know of now. Yeah.
Raptors didn't have wings, though.
What's that?
Raptors did not have wings.
Well, most raptors did not have wings, but there was a group of what are referred to as theropods from raptors that did have wings. Some examples of species are Archaeopteryx. That was like the first identified winged dinosaur. But there are a couple other species as well. It's actually why some birds are still classified as raptors. Hawks, eagles, falcons are in the raptor family. But yeah, this would be kind of a drastic bottleneck effect at the community level, where you have many species and only one persists. Usually you can apply this at the species level, not multiple species, but in one species. You could have an instance where a catastrophe knocks out most individuals and what is remaining. It doesn't always have to be an asteroid impact. It could be stepping on 5 bugs. You know, if that's the whole species of that bug, the bottleneck effect would be the very few that remain from that event. So when you see an example that sounds like some sort of catastrophe or disaster, think bottleneck effect. Yeah, but basically the similarity would be, you know, we always think of, well, birds don't look anything like dinosaurs. Well, a lot of evolution has occurred since then, but you can also imagine a situation where all mammals got wiped out, and what if the only thing left were bats, right? You know, it wouldn't paint the whole picture of, well, not all mammals look like bats, right? But that they just happen to be the ones who made it. Yeah?
Did they say, like, I read that there was an extinction period that was much larger than the dinosaurs before.
Yeah, so the dinosaurs are just kind of the textbook example that are used, because the dinosaurs were they dominated the earth and then they didn't, right? But there have been multiple extinction events. There have been even some in our own human history where humans almost didn't make it at all. I think it was 10,000 years ago, no, 100,000 years ago, the population of humans, even though we were already spread through much of the old world, and there were instances of evidence for humans already being in North and South America at this time, but there was a huge supervolcanic eruption that whittled the population down to about 10,000 or so. So humans almost early on in their evolution didn't make it. But we kind of restarted from those 10,000 individuals that remained. Yeah?
Is hunting always considered a bottleneck effect?
What's that?
Hunting?
Hunting. Yeah, hunting could be a bottleneck event of our own kind of demise there, right? So we can be that catastrophe sometimes. So think of catastrophe, disaster, and death, right? Yeah, so hunting may not be a natural disaster. You may not think of it as a catastrophe, but overhunting can certainly lead to these bottleneck effects. The example I like to give is actually regarding the great egret, egrets in general, but the great egret kind of got it worse. You maybe don't know that species off the top of your head, but if you just Google it, you'll probably have seen it even along the bayou here, these great egrets.
I've seen one of those.
Yeah, so they're all over the place now, but early in the 1900s, they went down to about a population of close to 100 only in all of Earth. The reason was because people at the time in the 1800s really fancied feathers in their cap. So they'd hunt the egrets just for feathers and hats. Complete waste, but we hear of kind of wasteful use of animals to this day, right? Shark fin soup is where they essentially take the fin from the shark and leave the rest in the water, in many cases, not all cases. Great egrets, they'd be killed just for a few of their feathers, and so this overhunting of the egret almost wiped out the population. Now they've rebounded and they're all over the place, right?
Same for rhinos. They're extinct.
What's that?
Rhinos.
Rhinos, yeah, poaching and people wanting the horn, right, of rhinos. Lots of large mammals. So, even before our more recent history, it's believed that most of the large mammals that were here in the New World, woolly mammoths and mastodons and the like, might have been, and giant sloths were kind of hunted to extinction by, cavemen, essentially at the time. So we wipe out a lot of things, but it's not all just recent. We've been doing it for a while. So yeah, those would all be examples though of bottleneck effects if the species makes it, right? It's not a bottleneck effect for a woolly mammoth because there's nothing left, right? So that would be the extreme. The bottleneck effect is just that fine point when you just end up with a few left. So bottleneck versus founder, catastrophe versus discovering something new. One thing that I feel like we didn't spend too much time on the forms of reproductive isolation. I like to characterize this as kind of one point along a timeline where reproductive isolation can occur. And I like emphasizing this point because I know there are a lot of potential vocab terms that are brought here, but basically everything can be summed up as being occurring either pre-zygotically or post-zygotically. So when you see an example of, what type of reproductive isolation does this example describe? First, ask yourself, is this pre-zygotic or post-zygotic? What that means is it happening before or after the fusion of two gametes and forming what's called a zygote? So egg and sperm, before they fuse and make that what we call a diploid organism. We'll get more into the details in later modules, but this point is kind of key, where you can have reproductive isolation form up until the point where the sperm happens to fertilize that egg and form the zygote, right? So if it happens before this point, we call it a pre-zygotic form of reproductive isolation. If it happens after, it's post-zygotic. Now, what happens, the sort of key word you can look for to determine if it's post-zygotic or not is hybrid. Because once a zygote forms between two species, you officially have a hybrid. It could die right there, but the fact that the sperm made it to the egg, even if just for a second, we would call that a post-zygotic state. Before it, this is everything leading up to that point. So you can have some things that are obvious, right? The individuals don't even live on the same continent, or they live on the other side of mountains and never experience each other. We'd call that habitat or geographic isolation. Due to just never seeing each other, they can't ever reproduce, right? So that is a pre-zygotic form of reproductive isolation because they can never even get to the opportunity to mate. So that's where space can kind of play an important role. Time, we live in space and time. Time can also be that factor. So one example that I like to give are multiple species of frogs. They'll live in the same pond, but they only mate at key times of day. So some frogs will mate during the day, some at dusk, and some at night. But they won't overlap in when they mate. And so if you're only ready to mate at night and there's another population that only mates in the day, then you're never going to mate because, even though you live in the same space, because of time. So we refer to this as temporal isolation, time being the factor here. It doesn't have to be that close in time. Seasons, when we think of plants growing in different seasons, other form of reproductive isolation. Plants that flower only in the winter versus the summer are never going to be able to mate because they just produce their gametes at different times. Some forms of isolation are behavioral, and they're really strong behaviors. So this is where things like mate choice come into play here. Maybe you have an individual that will only mate with a member of the opposite sex that is one coloration, or that performs some behavior. And then those individuals that don't have that trait, they won't mate with them. So one of the kind of hallmark examples of this are cichlids in Africa that live in Lake Tanganyika and Lake Malawi, cichlid fish that have evolved all in the same lake, but due to preferences, they've speciated into other species. And it's all based off of mate choice, where female cichlids will prefer mating with one color. And that preference is so extreme that they'll only mate with, say, a red male cichlid, and other females will only mate with blue cichlids. You let that happen over enough time, and you end up evolving these red and blue different species. They're behaviorally isolated. So due to simply not wanting to mate, reproductive isolation forms as a result. Now, the two that get we're getting closer to pre-zygotic barriers to this zygotic divide are mechanical and gametic. So sometimes, you know, they're in the right place, habitat, in the right time, temporal. The female even accepts the mate, but maybe their machinery isn't kind of too in line. Maybe the male even inseminates the female, but the semen never fertilizes the egg. Due to anatomical or physiological reasons, they mate, so we would call this post-copulatory, but they never form the zygote at the end. So we call these instances mechanical and gametic forms of isolation. So they're related off of each other, but a little different. Mechanical would be anatomical differences keep them from being able to mate. Gametic is they can even inseminate, but it'll never form a zygote. Yeah.
Mechanical, is that like the example in the book of the snail?
Yeah, so there are some snails. The example I like to give are with ducks, different species of ducks. So ducks, if you haven't, I mean this is biology, but ducks have very weird and reproductive anatomies. Basically, the penis and vagina of ducks are like corkscrew shaped. And males, it gets weirder, yeah, males have essentially the equivalent of innies. So when it's time to mate, their penises will inflate with their blood and kind of inflate like a corkscrew balloon. And then that will essentially, they have what's called an explosive semination. So once that happens, the semen will come out. But different species of ducks have different kind of corkscrew shapes. And so one form of mechanical reproductive isolation that keeps females from hybridizing is that they'll have a spiral that's maybe running clockwise, whereas the male might have a counterclockwise penis. And so they are mechanically isolated because the male can never actually inseminate the female in that way. So that's where the anatomy keeps them from being able to actually form a zygote. The gametic is kind of the next step. The machinery works, but something about the sperm doesn't, can't fertilize the egg. So even though they mate, even though the male has ejaculated, the sperm and egg don't fuse due to some chemical reaction or some physical barrier. After that, say the sperm makes it into the egg, then we're in hybrid territory. And this is all post-zygotic. So what can happen is one of three main things. So a lot of it depends on how distantly related are these populations. How long has it been since they kind of rejoined contact? So maybe you can think of an example where two species, we'll just use salamanders because this is a real example, but say two salamanders grow on one side, on separate sides of a mountain. They've been doing this for thousands of years. So because of this, because of differences in the environment they're experiencing, it's desert on one side and kind of more humid on the other, these little differences in environment result in selection for different things. As a result, the salamanders start to look a little differently, and you can actually see this. This is a real example in California, where along a mountain range you see salamanders look different on one side where it's desert versus the other side, which is the Pacific coast. Well, after thousands of years, say they kind of keep venturing south and eventually the mountain range runs out and they hit a point of contact. Well, they've been experiencing different things for thousands of years, so the question remains, are they going to be able to mate? If they mate, what will the population look like as a result? And so if it hasn't been long enough time, genetic differences haven't accumulated, then they could mate and you could get what we call fusion occur. So fusion is where two once hybrid populations, they make contact once again. They've been separated in some way, but now they've made contact. And now those hybrids are perfectly viable. So now they start mating with each other once again. So what happens if you start mixing, say you have a population A and B, what happens if you start mixing those genes again? What do you think happens to that population? Population A and B have been separated by a mountain, and then when they rejoin contact and they mate, and it hasn't been long enough, you start getting individuals with genes from A and genes from B, right? And they start mating with one another as well. Well, if the genes from B and the genes from A are mixing in a population, we just call that its own species, right? So now we have a population of mixtures of A and B. We don't have two species necessarily anymore. Maybe up here they still are. But down here, the genes have been mixed back together in this gene pool. We call this fusion. Fusion, literally fusion of the A and B genes. So if these individuals start kind of venturing back north, they're going to mate with their own species again. And so now these populations over time might get a little more A in them. And this A population might also get B in them. Give it enough time, and now A and B is just kind of all intermating with one another. And that's the biological definition of a species. So where we once had species A and B, now we have one population of species AB, you could call it, a hybrid species. And so this would be the result of fusion. The next thing that could happen is, I'll kind of go to the next extreme, which is reinforcement, and then come back to stability, because it's kind of an intermediate. So reinforcement is, say enough time has passed. Maybe now instead of 10,000 years separating these populations, they've been away for millions of years. And now, they don't even necessarily look the same. Maybe this population has lost arms and everything. So and they look almost like a snake at this point. Whereas these kind of look more salamander-y. Crappy salamander. And they, so now enough time has passed, now when they re-meet, maybe they're close enough that they try to mate, but the hybrids never really make it. You end up with, genetic freaks who just, have nubs, so they can't slither in the ground and they can't walk either. And so the hybrids just break down in some way. So either due to infertility, they can't, they've evolved so separately that they can't even mate, or through hybrid inferiority, hybrid breakdown, where maybe they mate and produce something, but that thing never really lasts. We would call this reinforcement, because what's happening is now that these populations have come back into contact, the reinforcement that's happening is selection acting against the formation of hybrids, because producing a hybrid in every instance is going to be a maladaptive situation, right? If you're one of these B species and you try mating with A, there's never going to be a good outcome. You're going to either produce non-viable offspring, the mating won't even make a zygote, or you produce offspring that just can't live long enough to reproduce themselves. So now selection starts acting against that. So you'll see behaviors develop that reinforce this idea of selection for behaviors that keep species B from mating with species A, or the other way around. So reinforcement usually results in maintaining species A and B as a result. So you can get species A and B fuse and form a hybrid species. You can get them to kind of make these awful hybrids and selection against them, which maintains species A and B. And then stability is really just the middle ground. Maybe you produce something that happens to do well in this environment. So maybe you do form this AB hybrid species, but every time it kind of goes north, it's in an environment it can't tolerate. Every time it goes, to the other side of the mountain, it's in an environment it can't tolerate. So selection is fine with hybrids existing right here, but they don't do well here or here. So now you get instability, almost the greatest biodiversity, because you end up with essentially what might become 3 species. Species A, species B, and now the hybrid species.
That's the hybrid zone.
Yeah, so a hybrid zone. So what I used to study was a population of fish. Xiphophorus malinche, which liked cold temperatures. They'd have their sword tails, they have swords here, but they generally liked relatively cold temperatures. I'm trying to draw a snowflake here.
Beautiful.
Yeah, what is this? Yeah, that's a snowflake, so it's cold. And then there was another species, Xiphophorus birchmani. There is another species. They don't really have the sword all that much. And they like, I'm just going to write hot. They were their own species, Xiphophorus malinche. They are their own species, I should say, Xiphophorus birchmani. And in between, they hybridize. And the hybrids can persist over time. So a malinche will mate with a birchmani and produce a malinche birchmani hybrid. And you see hybrids in these intermediate sections where it's not too hot, not too cold. But you go further north, the hybrids you don't see anymore. You go further south, you don't see hybrids either. So this would be potentially a case of stability, where we have all 3 possible outcomes occurring. You have the 3 species in a way. So stability is sort of the intermediate of reinforcement and fusion there. Any questions about this hybridization and how it relates to reproductive isolation? All right, so hybridization is a postzygotic occurrence. The zygote has been made. The hybrid is that zygote that's made between 2 species. One thing that in the past has often been not confusing, but sometimes overlooked, this idea of acquired versus inherited traits. I think it's overlooked because sometimes you'll read a question and it's describing a scenario and you're looking at the question like a giraffe stretches its neck and it acquires a longer neck over time. And so, your friend tells you that this is how they evolved. They'd stretch their necks, and then over time, the giraffes would get longer and longer necks as a result. Well, you're thinking the whole time as you're reading that about the giraffe, but there was a key word there about acquiring a trait versus inheriting it. That really is kind of the deciding factor in this question. So if you see the words acquiring or inheriting, these are really key words that you want to pay attention to. Yeah?
Acquired is kind of like use and disuse, like Lamarck, right?
Yeah, so acquiring a trait is just obtaining a trait through non-genetic means, right? So the example I give is a bit more sadistic than with the giraffes, but people get injuries all the time. So my uncle, for example, when he was 6 years old, a friend shot a rock through a slingshot, knocked his eye out, so he has one eye now. Now, that's a trait he acquired, right? When he reproduced with my aunt and generated my cousins, we wouldn't expect my cousins to have one eye, right? Because my uncle still got the genes for 2 eyes, right? They inherited his genes for 2 eyes. They might have inherited his stupidity, and maybe they're going to lose their eye again, but that hasn't happened yet. But that losing an eye, developing a scar, losing a limb or a digit, these are all acquired traits. So your phenotype is that you have 1 eye or 1 arm or 9 fingers, but unless it's genetically determined, it's not going to affect the way your offspring develop. So that was kind of there was this idea of, you know, the giraffes stretching their necks and doing that over generations and generations. Eventually giraffes just got bigger necks. But, you know, this description was about acquiring a trait. You know, they spend their whole lives sticking their necks up and their necks just get gradually longer. This, we now know, is not the case, right? You have to inherit those genes in some way. So acquired versus inherited. If you see these words, it's going to be kind of buzzwords that you should, you know, if you see the word acquire, you should think, okay, this isn't necessarily about the giraffe itself. This is, you know, a question about how evolution occurs through inheritance, right? Which ties into Darwinian fitness, how traits might evolve. And when we think of Darwin, don't think necessarily just evolution, think natural selection, evolution by natural selection. So he's the one who came up with this idea that it's really a numbers game. A numbers in terms of what?
Who's going to acquire?
The number of
Offspring.
Offspring, yeah. So how many individuals are you going to reproduce? How many copies of your genes
Oh, this is
are going to be generated?
relative fitness.
Yeah, this idea of relative fitness. It's all going to be about reproduction. So what matters is not how long do you survive. Survival can be very important, but it's useless if it comes at the cost of reproduction, right? So reproduction is really the importance here. I think I have driven this point home more, but know which traits correspond to which form of evolution. Convergent and divergent, and analogous versus homologous characters or traits. Right? So homologous traits are ones that have undergone divergent evolution. Analogous traits are ones that have evolved through convergent evolution. One letter game that I always think of that helps me remember this is convergent versus divergent. Convergent appears first. Analogous versus homologous. Analogous appears first. So A goes with C, H goes with D. If that works for you, then good. Otherwise, half of y'all probably have no idea what I'm saying.
I have a
right?
question.
But yeah.
Analogous.
To each other, then, well, then A is to D, so.
Right, so when you look at this, convergent goes first, so it's in the 1 position. Divergent is in the 2 position. Analogous comes before homologous, so it's in the 1 position. Homologous is in the 2 position.
Analogous just means same function but not the same structure, right?
Yeah, so it's like an analogy, right? They're not the exact same, but they get at the same point. So a sugar glider and a flying squirrel, they're not the same, but they both achieve the same thing. They can float from tree to tree. The same thing with wings, right? So dragonfly wings are not the same as bird wings, but they both fly. Homologous traits, they are evolved from the same ancestor, but diverged in what they ended up becoming.
Which is why they have the same structure but different function.
Yeah, so like the fin slash, so like the dolphin flipper slash human arm slash cat forelimb, all evolved from the same set of bones, but due to divergent evolution, evolved completely different functions for those traits. This one should be on y'all's radar since it's due tonight. So hopefully you won't need too much of a refresher between now and unit exam 2. But Hardy-Weinberg, with the exam, it won't be this won't be too much of an issue. But for the QR assignment, the point I'll drive home here is, especially read the questions, see if they're asking you for what percent, how many. So if it's how many squirrels, you want a whole number. If it's frequency, then you want a number between 0 and 1. And if it's asking what percent, you want a number between 0 and 100, right? So if it's saying how many, give a whole number, what percent, 0 to 100, what frequency, 0 to 1. So hopefully y'all will all be masters of Hardy-Weinberg, and those questions you'll be able to answer in a breeze. There are, I think, 2 or 3 Hardy-Weinberg equations, so you can use calculators for that part of the exam. But the numbers are pretty easy. Sympatric versus allopatric speciation. Not too much to say about this, but sympatry, think same. So they're in the same location, sympatry, same. So speciation occurring in the same location. So for example, with those cichlids that became behaviorally isolated, they all lived in that same lake in Lake Malawi, but due to behavioral differences, they speciated as a result due to differences in female cichlid mating preferences. Allopatric speciation, think back to the stickleback. So they had the spines, these the original saltwater loving sticklebacks, but the ones that were isolated in these glacial lakes, this fresh water, experienced different predation, and so they lost those spines, the stickles to their stickleback. But it only happened because they were largely separated geographically. They were allopatrically speciated. So allopatry is different locations, sympatry is same, same. And then, once you get speciation occurring in, at a larger scale, speciation events lead to something that looks like a phylogeny. Each one of those branching off points, each of those nodes represents a speciation event. So we can break that phylogeny down into history, right? So if we go from, along a timeline going to the present, we would say, looking at that whiteboard, that the population that would become species G separated from the population that would become species A through F. Then the population that would become species F separated from species A through E, so far, so on and so forth, until you have the last speciation event, the divergent evolution of species A and B, that last speciation event. But each node in a phylogeny represents some speciation event. And then just like I talked about at the beginning of class, monophyly versus paraphyly versus polyphyly. You can even type this out, and it's probably going to be in the first 5 things if you start typing monophyly. And then there are a lot of images that just drive the point home. So polyphyly is picking and choosing from different branches. Paraphyly is kind of snipping the branch that you've cut off, cutting off twigs that you don't like. And monophyly is, taking everything from that point you cut along the tree of life. So if you cut here, it's everything downstream of it, all the leaves and twigs that stem from that point that you cut at. But there are a ton of figures that just drive this point home. Paraphyly, polyphyly, monophyly. Monophylies are the most biologically accurate because we like to group things according to monophyletic groups. Protista is that one kind of biological exception where they're just the junk drawer. But everything else, genus, family, phyla, those are all monophyletic groupings, those clades.
I have a question.
Yeah.
So do we need to know how to draw the tree?
You don't need to know how to draw the tree, you want to know how to interpret it. So if you see a phylogeny like that, if I were to ask you, which species is most closely related to species A and B, you should be able to answer that from that phylogeny.
Mm-hmm.
If I were to ask you which species is most distantly related to species A and B, species D, E, or F, you should be able to answer that.
Oh.
But yeah, you won't have to draw a phylogeny. On the the topic of phylogenies, the synonymous terms are cladistics, cladograms, and clades. So cladistics is sort of the field or the method. It's how you make what's called a cladogram. Cladogram is essentially a phylogeny. So cladistics is the method used. Y'all did cladistics in this first activity. Y'all compared those amino acid sequences, right? So that was cladistics, which we used to make a cladogram right there. And then a clad is kind of the unit of a cladogram. So a monophyletic group. So species A, B, and C would represent one clade, for example. They're a monophyletic group. Species D, E, and F, we would say, is not a monophyletic group. So let's just use some examples here. This would be the example of a monophyletic group. If we kind of just took right here, because we took at one point of the phylogeny and included everything that came after. So we would call this a monophyletic group, which we could also call a clade, a clade. This, if I took this grouping, D, E, and F, would this be a monophyletic group?
No.
No, because we're not also including everything that came downstream. We're not incorporating this branch, which is still connected to this branch, right? So we would call this instead a paraphyletic group. Probably wouldn't be called its own clade, its own class. And then lastly, polyphyletic groups would be where we kind of just pick and choose. So if we took species A and B and just kind of picking randomly along that tree. So monophyletic groups are clades. You find a clade within a cladogram, which is made via cladistics. And then lastly, know your species concepts. The most important one, the most widely used, is the biological species concept, which relies on reproduction. Does it reproduce? If two populations reproduce, two individuals from different populations reproduce, we consider that one species. If they don't reproduce, they don't produce viable offspring, they're not a species. So, in biological species concept, reproduction is kind of the key. It's the gold standard, but it has its limitations. You want to be familiar with what the limitations of the best species concept, biological species concept, what those limitations are. Can anyone think of any right before we go here? What might be a limitation of the biological species concept?
The viable offspring?
So viable offspring.
The lack of viable offspring?
Well, so that would essentially define a species, right? So.
Are you talking about like the temporal, the hybrid breakdown, this concept?
Well, so not even that. Can we apply the biological species concept to all species? No, we don't know how, for example, if we found a fossil of, a Stegosaurus and then another fossil of Kentrosaurus, a different dinosaur that kind of looks like a Stegosaurus but with spikier plates, we don't know if that's a species because we can't get them to mate, right? They're fossils. So we can't apply the biological species concept to everything. We can't apply it also, that's an example using the fossil record. We don't know what in the past could or could not reproduce. There are some things in the present that we can't apply the biological species concept to. And that is, what about bacteria that don't reproduce via sexual reproduction, right? Two individuals may not ever mate in some species of bacteria. They all reproduce by binary fission. So how do we distinguish between one bacteria and the other? By the biological species concept, they are essentially different species because they're never mating with each other. They just bud off of themselves. But we still, call it all E. coli, right? So there's something beyond sexual reproduction that keeps it as a species. And so the biological species concept can't be really applied to those cases either, cases where sexual reproduction does not occur. And that, there's some other examples described in your book, but those are the two main ones. And then just be familiar with the other one, but they're not as important usually.
Sorry, do they all relate to both sexual and, sorry, asexual and reproduction?
For biological species concept?
For the polyphyletic or the phylogenetic?
So the phylogenetic species concept is based off of, it's how we relate fossil records. So fossils, it's not based off of reproduction because we don't know how they reproduce.
Morphological and ecological.
Yeah, morphological is like, how does it look? Do they look similar? And ecological is, what do they do? So do they function the same way in their environment? This is the least used. There aren't very many instances where this is applied.
Okay.